Good afternoon and welcome to another Research in Action brought to you by the Division of Research at Florida Atlantic University. My name is Karen Scapinato. I will be today's moderator. And before we get started, as always, a little logistics. We welcome your questions. If you want to ask a question, please hover your mouse in the bottom of your screen. You will see a little Q&A button there. If you click on that button, a little pop-up comes up and you can type your question there. You can type your question at any time during today's presentation. We will get to the questions at the end of the presentation. If we should run out of time and have questions remaining, we will ask today's speaker to answer those questions offline and post those together with a recording of today's presentation on our website. With that, it is my great pleasure to introduce today's speaker, Dr. Paula Kurdziel. Paula is a postdoctoral fellow in Dr. Randy Blakely's lab in our Brain Institute, which is on our Jupiter campus. And she works on intricate connections between brain and the peripheral immune system and completed her Ph.D. in genetics at Michigan State University. I look forward to hearing more about the brain and our immune system. Welcome, Paula.
Thank you so much, Karen, and thank you everyone for being here. So without further ado, we'll go ahead and get started. So today I'm going to be talking to you about your immune system and your brain and trying to really understand whether or not there is some degree truth in the statement that your immune system might actually be boss of your brain. So let's talk about your immune system. It's composed of cells and signaling molecules inside of your body that fight off infections. And it can even recall past infections so that we're less likely to get sick again with the same thing. Your immune system protects you from really any foreign things trying to enter your body. The hallmarks of an active immune system are typically things that we do not enjoy. Some of them being inflammation, which we can observe as redness and swelling. We can feel pain. We can even lose tissue function if we have an infection at a specific site in our body. These things are not actually caused by the virus or bacteria that might be infecting you. Inflammation is actually a really normal thing for your body to use to combat infections. So all of those symptoms that you get from being sick, those are actually because your immune system is doing its job. Imagine you get a cut and it's not washed quickly enough. So bacteria that might have been on your skin, which is typically a barrier for those bacteria that they can't get past, now they have an opening. So they enter through the cut, but thankfully your immune system is immediately alerted. It tries to fight off those bacteria by causing inflammation. The call or signal for inflammation is actually physical molecules. It's a bunch of molecules that are collectively called cytokines. Cytokines are produced by cells that encounter these bacteria, and then those cytokines get released from those cells. Almost like a call for action. The cytokines can be read by important immune cells like leukocytes or white blood cells, which can then move to the site of infection and help battle the bacteria. One of the major cytokines that we recognize is called interleukin-1 beta or IL-1 beta. And this cytokine is recognized by a unique receptor. And when a cell recognizes the presence of IL-1 beta, it'll cause this target cell to go ahead and make more IL-1 beta so that it can continue to pass on the signal for inflammation. This process is collectively called your innate immune system. This is really the first line of defense that your body has against any foreign cells that enter it. You may have also heard of a second step that your immune system takes, and this is in the development of antibodies and specific cells that guard you against future infections. This second part is called your adaptive immune system, and that's where all of those vaccines are targeting with an effort to really protect you from getting sick from those viruses or bacteria even in the first place. For the remainder of this talk, we will go ahead and just focus on the immediate steps that your immune system takes to fight off infections, which we call this innate immune system. For a moment, recall a recent illness that you might have had. Really, to fight off any infection, your body needs to do a few important things. You might have developed a fever. You're likely not enjoying a lot of food, or you have lost completely your appetite. You might be experiencing an increase in fatigue, which is really just forcing you to rest and nap more frequently. And you're definitely not in the mood to enjoy time with friends, nor should you, because you probably don't want to get them sick either. All of these changes are actually due to changes in your brain. There's constant communication between your immune system and your brain. Your brain actually can recognize when an infection is being fought in the rest of your body. Now, your brain, it dictates your behavior. So changing the ways that you eat or sleep, or your mood, or your interest in social settings, those are all changes in behavior. Believe it or not, but there's also a region of your brain called the hypothalamus, and this is responsible for the development of your fever too. You basically have like an internal thermostat located in your brain. Now let's think about these behaviors a little bit more in depth for a minute. Changing appetite, increased fatigue, social withdrawal. Sure, these are all pretty relatable when we think about being sick, but someone may be experiencing these symptoms unrelated to an illness. And if you think about it, these same behaviors are seen in individuals that are actually experiencing certain psychiatric disorders, like depression. So observing changes in behavior is actually the only way that we are able to diagnose someone with a psychiatric illness. So now seeing it in this light, it might not be so surprising to think about these behaviors stemming from changes in your brain. And it also really kind of begs the question about the connection between the immune system and the brain. There are definitely similarities in behavior that we see between physical illness and mental illness. Some studies have shown that there's actually a high rate of comorbidity between psychiatric and immune disorders. So comorbidity is just the diagnosis of two conditions or diseases in one patient. For example, in an assessment of blood of depressed individuals, they found that there were increases in cytokines, those molecules that signal for inflammation. Here, we're just going to refer to them so that you're reminded that they are pro-inflammatory. Another study found that antidepressant treatment, so drugs that aid in the ability to kind of alleviate some of these depressive symptoms, can actually decrease the levels of these cytokines in some individuals. It really kind of suggests that the source of the depressive behavior, at least in some patients, and the way that they experience it may actually not stem from just chemical changes in the brain, but it also related to an overactive immune system that creates too much inflammation. In another study, patients who were not depressed, but were instead diagnosed with cancer or hepatitis C, they're given a treatment for their disease. And this treatment is called interferon therapy. Well, interferon is actually another pro-inflammatory cytokine. So patients that undergo this therapy, it introduces really high levels of this cytokine into their body with the hopes that that's going to really help with fighting off the infection or disease in the for cancer at least. This therapy can actually cause about 40% of these patients to develop depression. And maybe like unsurprisingly, the depression goes away after the cytokine treatment is withdrawn. On the other hand, patients diagnosed with a chronic inflammatory condition like rheumatoid arthritis are actually more likely to develop depression, about 4 times more likely to develop it compared to the general population. Conversely, depression has also been found to be a risk factor for developing rheumatoid arthritis. So there's this really interesting two-way communication between your brain and your immune system. And in these studies, it really suggests that dysfunction in one of them could lead to dysfunctions in the other. This has caused scientists to develop a unique hypothesis for the development of depression that includes the immune system. Likely, this is not occurring in all individuals diagnosed with depression, but it may cover a subset of them that would then open them up to different types of therapies that might more directly target their immune systems. So it all starts with activation of your innate immune system. We've talked about how this happens when you get sick, But it can also happen under stressful conditions too. So too much stress might begin to signal your immune system to activate. Your innate immune system, like I've mentioned, produces these cytokines that signal for inflammation. I briefly mentioned one of them, IL-1 beta, which will come up a little bit more in this talk. But there's basically a cytokine storm anytime your immune system gets activated, and it produces a lot of different cytokines. Some that you may encounter if you decide to look into this further might be interleukin-6 or TNF-alpha. So your active immune system is going to produce all of these cytokines, and it's because it's basically a storm of cytokines. The hope is that they can cover a lot of ground when they're trying to eliminate and stop a pathogen. The brain's able to actually sense these changes throughout your body. You can kind of think of your brain as a master regulator. It's really in charge of nearly everything in your body, not really just your mind and your thoughts. Your brain also actually controls your breathing and heart rate. So it's always really surveying your body and making changes depending on what you're experiencing. This happens, and the brain is able to sense these cytokines, and the normal response is to release stress hormones. So this can cause alterations in the brain, it can cause changes in brain chemistry, and ultimately lead to the changes in behavior that I mentioned when you get sick. However, if this is happening too frequently or for longer than the actual length of an illness, then this might actually lead to depression. Now, depression is just one of psychiatric disorders, but it is one of the most prevalent ones, as you can see here. There are other studies that have been looking at the various psychiatric disorders and starting to really hone in on patients that are exhibiting altered immune system function, whether this be an actual diagnosis of an autoimmune disorder or the presence of elevated cytokines in their blood. So it's also of unique importance that we focus more on understanding the connection between these psychiatric disorders and the immune system because of recent years since the beginning of the COVID-19 pandemic. We can see here in the yellow dotted line, the rates of individuals, US adults, showing symptoms of anxiety and depressive-like behavior before the pandemic started. You can see a really sharp increase in the years following the pandemic in 2020 and 2021 in both anxiety-like symptoms as well as depressive-like symptoms. So it's really unknown whether or not this might be due to actual COVID-19 infections, since we're talking about the immune system here, but it's likely a combination of that, as well as the social isolation and stress that was put on nearly every single person throughout 2020 and 2021. This, to us means that we really need to focus on understanding the manifestations of these symptoms and the disorders themselves so that we can try and formulate new types of treatment that address the issues that so many patients have been having that might be stemming from changes in their immune system. In an effort to do this, many researchers, as well as myself, are utilizing animal models of these disorders. We are able to use genetically identical cohorts of mice so that we can go in and examine and understand the processes before we try and understand all of the variability that is floating around in the human population. These animal models simplify things for research and are a fantastic starting point to begin to delineate the mechanisms behind some of the most complex processes, such as what's happening in your brain. And believe it or not, but mice can exhibit behaviors similar to depression too. Typically, when a person is diagnosed with depression, they're asked about their mood, and we can't quite perform that with a mouse. However, a proven medication that treats depression, which we refer to as antidepressants, can alleviate some behavioral changes that we observe in mice. One example of this is called the tail suspension test, which I'll show to you in the next slide as well. But here a mouse is temporarily suspended by their tail. It's not a very enjoyable position for the mouse to be in, so they try really hard to get out of it. A normal mouse will struggle, and they'll show this agitated behavior, which is a lot of just swinging back and forth, trying to get out of this position. But if a mouse is depressed, they're going to show less of this agitated behavior, and they're going to remain immobile for longer durations. In addition to that, when we have these depressed mice that are exhibiting this increased immobility time, and we treat them with antidepressants, they will actually resume their attempts to get out of the suspension, and their immobility time will in fact decrease. They'll spend more time trying to struggle and get out of this position. There are a few different ways that we can look at depressive behavior in mice. And here we're looking at 3 groups. So let's go over these groups so that we can understand these graphs. The first group is treated with saline. This is basically just a saltwater injection, and saltwater doesn't do anything to behavior, but they do feel a needle poke, which is going to be a really important control because we have other mice that are going to encounter a needle poke but are treated with something different. So this is going to be our control normal mice. Then we have our next group that received an injection, but instead of water, they received something called LPS. So LPS is actually a piece of a bacteria. It's not the whole bacteria. So the mice are not actually getting infected with anything, but it's enough of a piece of it so that we can actually mimic the bacteria and their immune system turns on to try and fight off the pathogen. The same exact thing would actually happen in humans if they were treated with LPS. The important thing, though, is that because this is not an entire bacteria, the mouse's body will actually just naturally remove it from their systems within about 24 hours or so, and then they actually go back to normal. So their immune systems are not activated for very long. Then we have our last group, which is UCMS, which stands for unpredictable chronic mild stress. This protocol puts mice through a few different stressors that can range from bouts of social isolation, which is not great because mice are pretty social creatures. They could have wet bedding in their cages or be exposed to loud noises. In general, these stresses are quite mild for mice, and just one of them wouldn't actually cause any depressive behavior. But these are unpredictable, and they're given for a very long period of time, hence the chronic portion of this. When this happens, the mice will exhibit depressive-like behaviors. The first of these behaviors we're looking at is called the open field test, or OFT. The mouse is placed in a rather large empty box, and they get a chance to explore and move around for anywhere between 30 and 60 minutes. You'll see here that a stressed mouse does not move around as much as a control mouse, dictated here, and they kind of hug the corners quite a bit. Remember, mice are prey animals, so they do have a natural tendency to avoid wide open spaces, which is why you'll see even our control mouse will kind of hug the corners more than exploring this open center here. But they're also exploratory by nature. And so they're often looking for food or shelter, which really causes them to just explore. They've never been in this box before, so they kind of want to go all over the place. You see here in our graph, this is going to be the border zone, or what we call peripheral, so outside of the center, everything on the edges of this box. And when the mice are sick or when they're stressed, they basically hug the corners of these boxes. In tandem with that, this is going to be looking at the time they spend in the center, and they just don't explore as much either. So this is going to be indicative of what we look at with either anxiety behavior or also depressive-like behavior in mice. Lastly, they had these groups perform the tail suspension test. And here we're looking at time immobile. So if a mouse is more immobile, then it's exhibiting more depressive-like behaviors. And our sick group and our stressed group are indeed immobile for long periods of time, meaning that they are not struggling as much to get out of their suspension. The researchers from this study also wanted to take a look at what might be changing throughout the brains of these mice. And here you'll see 3 different brain regions that basically assess all over the brain what might be changing. And so right here, we're going to go back to our inflammatory molecule, our IL-1 beta. And you can see higher levels of this cytokine in our sick mice as well as our stressed mice. And this happens throughout the brain. And you can see that in some brain regions, if the mice are sick, they're going to have higher levels of this cytokine, IL-1 beta, compared to our stressed mice. And that's likely because sickness does launch quite a large inflammatory response. Now, if you've heard anything about depression, it likely falls in the realm of serotonin, which we're going to go to next. So serotonin is a chemical in the brain, and it's the target for most of these antidepressant drugs. Prozac is a very commonly prescribed antidepressant, and it affects levels of serotonin. In most depressed individuals, the changes in mood and behavior are likely due to less serotonin being available. So these same groups were assessed for their levels of serotonin in these same brain regions. And you can see here that the sick mice as well as the stressed mice have lower levels of serotonin. It looks to be not completely throughout the brain, but at least in the stressed group, we're seeing much lower levels of serotonin compared to our black bar, which is going to be our control mice. And when the mice are sick, it looks like these levels of serotonin do get lower, but it's not all over the brain. Other studies have shown that if you treat these kinds of groups of mice with antidepressants, that you can actually stop the depressive behavior from happening in these mice. And we'll go over a little bit how these antidepressants can elevate levels of serotonin in your brain. So serotonin is made from components that you actually get in your diet, like eggs, fish, dairy, and meat. They're really reliable sources for a precursor of serotonin called tryptophan. Once serotonin is synthesized, it can affect your various aspects of your behavior. It's most commonly known to regulate your mood, but it's also responsible for parts of your cognition. It also regulates your sleep-wake cycles, and it's really important for memory too. Serotonin can also be turned into melatonin, which many of you may know is also essential in helping us get to sleep. Serotonin is actually made by just a few pockets of cells in your brain, highlighted by these orange circles here. However, it gets released, as indicated by these red arrows, all over your brain. So that's really why serotonin can impact just a lot of different functions. Your brain really works by communicating chemical messengers, and serotonin is one of them. You can see the chemicals denoted right here. Cells will go ahead and release a chemical message in order for you to have thought and to be able to interpret language and allow for movement and a whole lot more. Here you'll see a nice diagram of these chemical messengers. And at this point, this is where two neurons or brain cells talk to each other. Well, the top one is going to be releasing our chemical message to another. And this bottom one is going to read or sense that chemical message. This chemical message here is actually serotonin. We can easily see this depicted in kind of a simpler diagram down here. This diagram shows a synapse, which is the point where two neurons meet. We have our presynapse cell before the synapse, presynaptic. And this is going to be our serotonin neuron here. 5-HT is going to be how we shorthand say serotonin. This cell will release serotonin. And this will be in a physical opening between the two neurons here. Serotonin is then recognized by the post or after the synaptic cell. And then basically the serotonin, once it gets recognized, it will need to be removed from this area. Otherwise, it'll keep signaling, and that's going to be actually what causes some aberrant behaviors. So what we have here is our serotonin transporter. So it transports our chemical signal, our serotonin, and back into the presynaptic cell. It's basically a vacuum for serotonin. So after the serotonin is released, this SERT, or the serotonin transporter, dictates how long the serotonin stays and can signal, and actually how much of it is able to signal. Interestingly, serotonin is not just a chemical found in your brain. In fact, about 95% of your serotonin in your body is actually made in your gut, and it exists outside of your brain. The serotonin made in your gut is important for regulating gut motility or movement, and serotonin from here can actually easily be taken up by the blood. There's not a lot of free-floating serotonin in your blood, though. The serotonin is immediately picked up by platelet cells in your blood because they themselves actually have a serotonin transporter, or SERT. Here, a vacuum for serotonin. Here, you'll see just a chart of a lot of different immune cells. Of importance is actually how many of these cells utilize and recognize serotonin. So those words in blue or red are indicative of cells in your body that recognize serotonin in your blood. This is important to note that the serotonin that's in your blood, it can't actually pass into your brain. So the stocks of serotonin in your body are going to be completely separate from your stock of serotonin in your brain. However, because we know that behaviors that are indicative of psychiatric disorders might actually be due to changes in the level of serotonin in your brain, if there's dysfunction at all in any of these molecules that are related to the serotonin system in your brain, very likely they're also going to be dysfunctional throughout your body. And thus you can kind of start to see the connection between dysfunction in the serotonin system in your brain and how your immune system might become dysfunctional as well. So dysfunction in serotonin signaling is implicated in many, if not all, of the psychiatric disorders, and definitely all of the ones that I have listed here. I'm going to, in the next slide, walk through briefly how one class of these antidepressants, which is maybe not surprisingly used as a first line of medication for many of these disorders. I'm going to show the target of these medications and how this target could also be implicated in changes in the immune system. Okay, so as a reminder, we have our synapse here. SERT being the major determinant of how much and how long serotonin is present and able to communicate with another neuron. A major class of antidepressants that is proved to relieve depression in many, but not all patients, is called an SSRI. This stands for selective serotonin reuptake inhibitor, and it basically blocks SERT. This allows serotonin to then communicate more with our postsynaptic neuron here. It's allowed to spend more periods of time in the synapse, and there's also more that can accumulate here if the serotonin transporter, or SERT, is blocked. The Blakely lab has studied for a while the effect that are in, and then this, like I mentioned, also raises serotonin levels and can alleviate some of the symptoms associated with depression. So we've also studied that the effect that the inflammatory cytokine IL-1 beta can have on serotonin signaling. And it actually turns out that SERT is a target for this. So this actually increases serotonin transporter activity. It effectively makes SERT into a super vacuum. And this is going to decrease the amount of serotonin that we have that's able to communicate to our postsynaptic cell here. This is what actually gives us many of the changes in behavior that we experience when we're sick. But it's also, as we're seeing, maybe indicative of the development of depression in some individuals. Studies in the Blakely lab have used a lot of different genetic manipulations to really try and understand the mechanism between how infection and sickness can impact depressive-like behaviors. So here we're taking a look at the tail suspension test again, which is a really key behavioral paradigm that we have in assessing behavior, depressive-like behavior in mice. And we already mentioned LPS, which is going to be our bacterial mimetic. So WT stands for wild type, which basically means that these mice are normal. You'll see that these normal mice have a reaction to LPS, So they have increased time immobile during the tail suspension task, thus indicating that they are experiencing more depressive-like behaviors. However, mice that have undergone genetic manipulation to remove a key immune receptor, and in fact, this receptor recognizes our inflammatory cytokine, IL-1 beta, they're immune to LPS. They do not show these depressive-like behaviors. Similarly, when we have mice that lack the serotonin transporter through genetic modification, they also do not exhibit the LPS change in behavior. Their time immobile is more closely resembling that of our normal mice here. So the serotonin neurons in the human brain are actually very similarly matched to the mouse brain. It further gives validity to our studies when we're using this animal model. So for both of our brains, just so we can orient everyone, the front part of the brain near the eyes is going to be towards the left for both our human brain and our mouse brain. And then the back of the brain is going to be towards the right here in our human brain and in our mouse brain. In the mouse brain, you'll see a cluster of ovals and circles towards the back of the brain, and this is where the mouse serotonin neurons live. They have really similar pockets of serotonin neurons to humans. And you'll see these red lines are also indicative of where serotonin gets released throughout the brain. You can see it goes nearly everywhere in the mouse brain as well. If we look at a slice of a mouse brain, you'll see a really distinct organization of these serotonin neurons, which are indicated by these dark spots here. They actually are organized in a pattern, and depending on where they're located in the brain, we can, studies have shown that there are differences in which parts of the brain they actually communicate to and release serotonin. So one of my main goals has been to understand which of these regions of serotonin neurons may be uniquely responding to the immune system. And if it's not unique, if it's all of them, also looking at what that might look like. Since we know that IL-1 beta is able to impact SERT activity, I began looking at which of these serotonin neurons were making the receptor or the sensor for IL-1 beta. Here in green, you'll see a very similar distribution of our serotonin neurons. Again, shorthand 5-HT for serotonin. In red, you'll see that these neurons are also expressing the immune receptor that recognizes IL-1 beta. And this third image is a merge of the two. And yellow will indicate whether or not if a cell is expressing both serotonin and the receptor. And you should start to appreciate here that not all of these cells are yellow. So it's not all of the serotonin neurons that are making this receptor. Thus, not all serotonin neurons are able to respond to IL-1 beta. However, we can start to piece together the puzzle using previous data that indicates what a specific portion of these serotonin neurons communicates to. And you can see here that there's a portion up here of these serotonin neurons that are very bright yellow. So we have very high expression of our immune receptor in this group of serotonin neurons. Previous studies have, as I mentioned, mapped the communication for these serotonin neurons. And that's been really used to determine which brain regions we should start looking at as being influenced by the serotonin neurons that respond to the immune system. So in here, I've indicated a few brain regions. We won't get into the specifics about these brain regions, but I'll be happy to take questions afterwards if you want some more elaboration. But brain regions that are receiving serotonin from these serotonin neurons that are able to respond to the immune system, they're involved in things like motivation, reward, and pleasure. There's also a few regions that are really key in memory formation and emotion, behavioral emotional behavior output. And so the last one that I'm going to talk about is a brain region that is involved in aversion. And so all of these regions, all of these brain regions very uniquely receive serotonin from serotonin neurons that are impacted and able to respond to the immune system. So the main goals of my research are really to further determine which serotonin-related brain circuits are being influenced by this immune receptor, which we call IL1R1. Additionally, what kinds of behaviors might the inflammatory cytokine IL-1 beta be influencing? And lastly, what normal function might be influenced by the presence of the cytokine, and what portion of this might actually be dysregulated during illness, both physical illness and mental illness. In order to answer these questions, we developed a unique mouse model that genetically removes the immune receptor only from serotonin neurons. Now, I won't go into detail about the specifics about how we constructed this mouse model, but if the mouse expresses an enzyme called CRE, then our serotonin neurons will not be making the immune receptor. Here you see these two images. Green is indicating another marker for serotonin neurons, and our immune receptor is going to be in red. You'll see in our mouse that is not expressing CRE that our serotonin neurons have expression of this immune receptor, dictated by these red dots here. When they are expressing CRE, we no longer see the red dots or our immune receptor in our serotonin neurons. So for the next few slides, I'm going to be showing you CRE negative and CRE positive mice. CRE negative are the ones that still express the interleukin-1 receptor or our immune receptor in our serotonin neurons, and CRE positive mice don't have it anymore. So previous data had shown that when we remove this immune receptor, we no longer get an LPS effect. And this is instead of looking at, although it does look like a very similar graph, instead of looking at immobility behavior, we're actually looking at a measurement of the serotonin transporter or SERT. So with LPS, we have our serotonin transporter taking up more serotonin. That's what our y-axis is telling us here. So this essentially is showing us that the serotonin transporter, when a mouse encounters an active immune system or inflammation, the serotonin transporter turns into a super vacuum for serotonin. When we don't have that immune receptor all over, so this is a whole body deletion, genetic deletion of this immune receptor, LPS is no longer able to change SERT activity, so it remains normal. Very similarly here, we have our mouse model. Remember, CRE negative is going to be those that still have our immune receptor, and CRE positive are the ones that do not have our immune receptor. When we have our normal mouse, which is going to be really these first two bars, and it's treated with LPS, our serotonin transporter turns into that super vacuum. It takes up more serotonin. When we only remove this immune receptor from serotonins, and this immune receptor is really present all throughout the body. It's a really key receptor for passing along the signal of inflammation throughout the rest of your body. But if we only remove it in serotonin neurons, then LPS doesn't have an effect either. So not only do we have changes in the serotonin transporter or SERT, we also have inherent changes in our serotonin neurons. So what we're seeing here is a group of females or a group of males that were treated with our saline, our control salt water, or LPS. And now we're looking at something different. We're looking at how many of these neurons were recently turned on. So when a neuron turns on, that's when it can send its chemical message. That's when serotonin is released and read out by another cell. So if these cells are not on, then they're not releasing serotonin. You can see here that in our normal mouse, in both our females and males, so our CRE negative normal mice, LPS tends to shut off these serotonin neurons. we're looking at here a readout for activation or turning on of these neurons, and there's less of it when we have LPS. So not only is our SERT, a super vacuum, sucking up more serotonin, but we also have these serotonin neurons not turning on as often. So really, it's a double whammy for losing serotonin levels in your brain. However, when we remove this immune receptor from serotonin neurons, LPS doesn't have an effect. It doesn't lower them, the levels of neurons turning on anymore, either in the female or the male here. So this is really important. Without this immune receptor, the serotonin neurons can still turn on as much as they're supposed to, and their serotonin transporter doesn't get turned into a super vacuum. So we've looked at this effect on whether or not other neurons are turning on throughout the brain. And we've been targeting regions of the brain that I mentioned are going to be actually receiving serotonin from this population, the ones that express this immune receptor. And so that's going to be portions of the hippocampus as well as lateral habenula. So hippocampus is our region that's important for the establishment of memory as well as learning. And the lateral habenula is our aversion center in the brain. These regions are also impacted by loss of the serotonin immune receptor. When we have a normal mouse, we see less activity. These neurons are not turning on as much when they encounter LPS. But if only the serotonin neurons don't have this immune receptor, then there's no impact in these downstream brain regions and they function as normal. So the next part is really trying to determine which behaviors are impacted by serotonergic immune receptor expression. So by honing in on places like the hippocampus and the lateral habenula, these brain regions are known to impart certain behaviors. So what we're really interested in looking at is targeting these serotonin cells and either turning them on ourselves or turning them off ourselves, and then looking at behaviors that are mediated by places like the hippocampus, which is important for learning and memory, or the lateral habenula, which is the aversion center of the brain. So we can actually test out memory of these mice when we either activate our serotonin neurons or turn them off. And then also importantly, we want to assess how are these serotonin neurons different from other neurons. You recall hopefully in this picture, not all of these serotonin neurons are expressing this immune receptor. So you could have a serotonin neuron expressing it, and then its neighbor right next door to it is not responding to the immune system. So what makes these serotonin neurons that can respond to the immune system, how are they different than the other ones? And then finally, determine whether or not these neurons can be uniquely targeted with a drug. Our main goal, our big goal, is to try and figure out if we can alter behavior by altering these serotonin neurons. So with that, I, of course, need to acknowledge everyone that made this research possible. Dr. Randy Blakely, my mentor, as well as various Blakely lab members, current and previous members, our joint collaboration with Dr. Ning Kwan and his lab as well as the various funding sources that allowed this research to be possible. So I thank you for your attention, and I'm very happy to answer any questions for you.
Great. Thank you very much, Paula. I learned a lot. I did not know that serotonin was on our guts to begin with. Just as a reminder to everybody on the call, if you have any questions for Dr. Kurdziel, please use the Q&A button in the bottom of your screen and you can type your question there. We're going to go through them as they're coming up here. So Paula, first question. You mentioned earlier that serotonin affects mobility and serotonin produced in the gut affects mobility. Can you go into a little bit more detail and address a little bit more on what you mean by that?
Sure. So it's really easy to think about it in relation to some disorders. So I'll tie it back to one of the psychiatric disorders that the Blakely lab studies pretty frequently, which is autism spectrum disorder. And so we ultimately think that there's the manifestation of these behaviors due to dysregulated or lack of serotonin signaling, right? And this can affect the repetitive and social behaviors that we see in individuals that are diagnosed with autism spectrum disorder. But in a very large portion of those people that are diagnosed with ASD, they also have intestinal and gut problems. So very frequently they're diagnosed with things like IBS. And this is because if we have dysfunction of the serotonin system in our brain that leads to changes in behavior, very likely you're going to get disturbances in your gut and also clearly in your immune system.
Great. Thank you. Okay, very important question here next. You mentioned that you're using rodents, and the question is related to the difference between a rodent brain and a human brain, and what type of rodents you're using, and whether they're specifically bred for the lab.
So we do have genetic models, like the one that I mentioned, where we can delete this immune receptor for serotonin neurons, and that's one that we do specifically make for our lab. But in general, we're using mice. Mice have a lot of advantages when we're doing research like this because we know the entirety of the mouse genome. So we're actually able to manipulate it using genetics pretty easily. And we can develop lines of mice that have mutations for us to be able to target something so specific, like an immune receptor, on just a tiny portion of cells in the brain. So while the strain of mice, when we talk about mouse research and neuroscience, typically they're all going to be genetically identical. So they're all inbred mice to try and really remove the variability. Like I said, one of the main reasons why we do mouse research is so that we can try and really get at a specific mechanism and try and understand things on a really simple level, because humans are very complicated and very variable. So it's really great for us to start our research by looking at something that doesn't have a lot of variability in it, which would mean looking at a genetically identical line of mice. And then we can go ahead and put mutations into them so that we're able to kind of narrow down our studies and have a more focused approach when we're looking at a specific molecule.
Great, thank you. And as you can probably already hear, any of these lab animals are specifically bred for the lab and not the animals that you would encounter anywhere outside in the world. So they probably wouldn't survive. Okay, as an extension though to this question, how does your research extrapolate to humans, and how do you think your research ultimately, what's your ultimate goal? The ultimate goal would be, I would assume, helping humans, right? And whether it's in depression, excuse me, or other conditions. What is your ultimate goal, and how do you think your research that is currently not done in humans could relate to human disorders?
Absolutely. So that's a huge importance. And hopefully I was able to show you in the beginning how this is impacted in the human population and what we're trying to really understand with our mouse lines. But essentially, one of the things that I wasn't able to address today in my presentation was the difference between males and females. So males and females have a significant difference in their diagnoses of psychiatric disorders. They also have differences in their immune systems, believe it or not. So we're really interested in understanding this group of serotonin neurons that are able to respond to the immune system. Are they different in male and female mice? And then if they're activated, do they show differences in behaviors as well? When these neurons, these serotonin neurons, are turned on, do we see difference in behavior between our males and females? So really trying to understand, is there some biological reason why we see differences in males and females and their brains and their diagnoses of psychiatric disorders and their responses of their immune system? So that's a big part of what I'm also studying.
Very interesting. Okay, coming back to one thing that you mentioned in your presentation, you mentioned eggs and seeds and poultry and a number of other things, soybeans that have precursors to serotonin in them. Can you go in a little bit more detail how the food and nutrients and even hydration impact the brain?
Sure. So just start off with serotonin. So your body doesn't actually make the precursor to serotonin. So we can only derive it from our food. So that precursor is called tryptophan. And we're coming up on Thanksgiving. So many of you might have heard that turkey has a lot of tryptophan in it, and that's what makes you sleepy. There's a little bit of truth to that, although really it's a little bit more complicated than that. But you do get a lot of tryptophan from turkey. So that actually is going to increase your serotonin levels. So it's really important for you to take in all of these things, a really balanced diet, because some of the building blocks for the chemicals in your brain are not produced by your body, and you actually get them from your diet. Hydration is also important, of course, for your brain, as well as a healthy diet. But in general, your brain, I think this also what it stems to is your brain is so densely packed with blood vessels. And so it actually needs all of the components from things from a healthy diet, all of that hydration in order to function properly. So it needs a lot of energy. It needs a lot of sleep as well. So all of these things are just in general, like when people tell you, what do you need for, a healthy brain? It's probably all of the things that you would think dictate a healthy lifestyle as well. And I think it's really key in remembering that some of the things in your brain are going to be dependent, like you need them there, and you're only able to derive them from a healthy, balanced diet.
And that's really where the recommendations for a healthy diet and healthy lifestyle are coming from, right? Because we are more and more recognizing what certain compounds and components of our diet do to our body, and what has a positive and also what has a negative effect. So that's really where there's ultimately those recommendations are coming from. They're based on the type of research that is continuously ongoing. Okay, Paula, coming back to the cytokine levels, can you address whether those levels vary throughout the day?
So they actually do, but in very, very low levels. So in general, your body produces a little bit of these pro-inflammatory cytokines. However, when you get infected or your immune system is activated, those skyrocket. You can start getting like an 80 times more than your body typically has in its system. However, the cytokines are there to a certain extent at all times, and then they do fluctuate throughout the day. So I'm not going to exactly recall when you have the highest levels of IL-1 beta, but because it can fluctuate throughout the day, it's actually important for normal function of you going about your daily life. So you need that fluctuation in very low levels of these cytokines to be able to function and have a good established circadian rhythm, for you to have good cell-to-cell communication. But yeah, they're there at very, very low levels, and they fluctuate throughout the day.
Great, thank you. Another, as a follow-up, another cytokine question here. Are there any differences between the cytokines that you mentioned compared to the IL-1 beta? Or do they all have the same main functions of signaling a stimulus throughout the immune system or in the immune system?
So they're really similar, but they're also different because it's always more complicated than anyone can get into in a short talk like this. But so they all signal inflammation. They're all pro-inflammatory. Whenever a cell recognizes one of these pro-inflammatory cytokines, it's going to try and make more of that pro-inflammatory cytokine to pass along that signal. So the signal of inflammation keeps getting passed on and bigger until ultimately your body has systems in place to shut it down when it doesn't need to be there. But these cytokines, they have different receptors or sensors on the cells. So if a cell can recognize IL-1 beta, which is the one that I talked about today, that doesn't necessarily mean that it can recognize IL-6 or TNF alpha or those are the other two that I mentioned, right? So it needs a very specific matched sensor on a cell to be able to be read.
Right. Yeah. Okay, coming back to serotonin, you mentioned, the effects that low levels of serotonin has on mood, for example. So the question here is, can you take serotonin? You mentioned that we do need to take up serotonin with our diet. We don't produce it ourselves. can you, if you have a serotonin-related disease, can an addition of intake, ingestion of serotonin counteract that disease and help that disease?
So you can't actually take supplements of serotonin and have it affect your brain. So if you ingest serotonin, very likely it's just going to stay in the rest of your body where serotonin still plays a big role, but it actually can't pass this barrier between your body and your brain. So this is called the blood-brain barrier, and serotonin's not able to pass through that. That's why when we consume foods that contain tryptophan can actually be passed from the brain to the body and all over the place. So that's what really helps us synthesize more serotonin. If someone has a disorder that unfortunately causes them to either make less serotonin or they have changes in their serotonin transporter, right? No matter how much tryptophan you might put in your system, your body might not be able to compensate, because there are going to be, if there's mutations that don't allow for a lot of synthesis of serotonin, or if your serotonin transporter is always going to be a super vacuum, then more tryptophan doesn't necessarily help in that.
Right. Yeah. So does the uptake of a precursor of a serotonin will only help if you still have a functional mechanism that makes serotonin out of the tryptophan in your body, and that's sometimes not the case. Okay, we're almost at time. I'm going to leave you with one more question here, and one more answer, I should say. I see we have a number of other questions, so Paula, we're going to ask you offline to answer the remaining questions, and we're going to post the answers on our website. Yeah, are there any thoughts on the contribution of continuous inflammation in the development of Alzheimer's and dementia if low serotonin affects memory?
So there's actually ongoing research in the Blakely lab that is investigating the impact that this immune receptor on serotonin can have in the development of Alzheimer's disease. So we have a collaboration with a group in Israel that's working on this. We have an Alzheimer's genetic mouse model, and then we have combined it with the mouse model that I showed today, which removes the immune receptor from serotonin neurons. Now we're trying to figure out if that can actually alleviate some of the cognitive and behavioral deficits that we see in our Alzheimer's model. So in Alzheimer's, they're finding that there's a lot of inflammation that's happening, and very likely it's due to just a ton of cell death happening. When a cell recognizes that another cell has maybe burst open and died next to it, that stresses that other cell out. And so they might start producing things like cytokines. So we're seeing that individuals with Alzheimer's, and in our mouse models for Alzheimer's, there's elevated levels of cytokines. And so we are on the road to try and understand what impact serotonin, and specifically this immune receptor on serotonin neurons, might contribute to those changes.
Great. Thank you very much, Paula. Very interesting research. Congratulations. Good luck with the remainder of your postdoc here. And thank you, everybody, for calling in. And we hope to see you next time again. Thank you, Paula.
Thank you so much.